So our next session, I'm going to bring to the podium Dr. Raphael Goradetsky, who's affiliated with Hadassah in Israel. The World Stem Cell Summit goes back a long way, and I've spoken at more conferences in 20 years than you can imagine. And one of them in London was on patent law and regenerative medicine and stem cells. And my plane landed, and I found out I was the first speaker in the morning. No time to freshen up. I walked right to the podium and gave a talk. And believe you, I was what they say, flabbergasted. I didn't know whether I was coming or going. But no matter, I was taken under the wing by another speaker there by the name of Raphael Goradetsky. Rafi and I spent the free time touring London together, eating fish and chips and mushy peas and chips and such. And I got to know this gentleman very, very well. He is a genius on mesenchymal stem cells, MSCs. He was a cancer researcher. He is part of the startup nation of Israel. And he flew over from on the outskirts of Jerusalem to be with us today. MSCs are critically important in the future of regenerative medicine and developing cell therapies. There are just a tremendous number of clinical trials going on. So Rafi is going to discuss with us today potent cell therapy in Israel with mesenchymal placental cells. Welcome, Rafi, to the World Stem Cell Summit.
So I take the opportunity of talking on our technology with placental stem cells or stromal cells in order to criticize a little bit the approaches that were taken with MSC so far. I began my recent career, I would say, 30 years ago by promoting fibrin-based matrices for cell growth and so forth, which eventually grew into companies, and it led me to mesenchymal stromal cells, which were called mesenchymal stem cells at that time. And I later on found out that the placenta is a good source of cells in the absence of other cells of interest. So let's continue. First of all, I wanted to thank everybody who gave me a hand in my last project on the placenta. Since at the end, nobody really cared what people say, so rather give it before. Well, let's go to the basics. But 20, 30 years ago, people believed that now that we have cells that we can grow and have mesenchymal stromal cells, which were called mesenchymal stem cells. Embryonic stem cells came out. We could mix some growth factors in and put a scaffold, and we can build everything. So we could build human beings, we could build tissues and so forth, and that was actually what people expected, but it didn't work out. Now the question, why wouldn't it work out? If that's what people anticipated at that time, at the beginning of the century, you can see that the expectations were sky high. We will build human beings in the beginning of the century, 21st century, biotech bodies and whatever. If you open futuristic articles, you get exactly the same articles. Nothing has changed since then. And there is a reason for that. So the first reason was that people anticipated that once the ban on ES cells would be lifted, then there will be fantastic flourishing of this area. When Obama signed the allowing funding embryonic stem cells, actually they thought it will open the field, and in years all these things will actually come true. This is, for instance, a presentation that I found about 30 years ago of what people expected. Now you can see that the right side, which is tissue engineering that people expected, you see the printing tissues and so forth is what people are doing nowadays. I mean, nothing has changed since then. The only really hint that this is old slide is the computer, because the computers have changed. This is computers from the 80s, and now the computers look different, but all the fantasies are the same, but were not really fulfilled. And the question is, what really prevented us from having tissue engineered? So I give you an example of the skin. There were a few companies, Thermograft, Appligraph, Ocel, that had artificial skin. It looked exactly like the skin with all the layers. But when they tried to use it, it wasn't so successful. What was missing? The blood vessels. So when you take a tissue, they build this three-dimensional tissue, whatever tissue it will be, eventually if you don't immediately revascularize it, it will die. I mean, you can't really expect that you have a heart patch, that you put it on the heart and can get revascularized eventually, but when you press on your own heart on the artery for 5 minutes, you killed a fantastic heart that works fantastic. So you really cannot expect that engineered tissue could work better than that. So tissue engineering, which was a great hype, hasn't really fulfilled the expectations. But people were really looking for having something that you could present. And even talk about my work. This is work on Calvarian defects and bones that we use our fibrin microbeads and mesenchymal stem cells. And when we implanted them, we really could see some bone formed. But this was a thin bone of 1 millimeter or so. But if you want to correct a human bone, you have to put half a kilo of cells with matrices. You wait 5 minutes, it's all dead, because it's not connected to blood vessels. So the only good results that we received in this area was a collaboration that we have with the FDI, with the Maxillofacial Regeneration Institute, in which we managed to build some cartilage. And the advantage of cartilage is that it's very poorly vascularized. So if you have good cells, good technology, you can get something out of it, but not the expectations of building body artificially. But the money was flowing to stem cells. People had to bring results somehow. They got funding. What would they share? What would they show? What would they share with the people? So they began to publish, and a lot of the publications were faulty. Even good researchers eventually had to back up from the publication, which were too optimistic. For another one that was actually a good example. But the worst example was by Macchiarini, who built tracheas, tissue engineered. He looked fantastically, implanted in patients. I don't know how he got the permission to do it. Karolinska is a good institute. It's not in Slovakia or anywhere else. It's Europe. And eventually killed all the patients. And it took a long time to uncover the horrible things that he has done. But how did all these things happen? Because people expected success. And if you don't have success, you invent the success in order to make it work. So, but the cells were there. So people began to inject them. For just injecting cells anywhere, and to the surprise, they got positive responses. So they invented stem cells therapy. You go inject the cells, you don't know what will happen. Eventually, you get some positive results, and you open institutes around the world. You call it the Swiss Institute, but you open it in Slovakia or places like that that have no regulation. And you bring some patients that say that they saved their life. And you have other companies in Jamaica that work in Jamaica. They work anywhere else, but they have clinical trials in Jamaica. They show some results, but also the FDA warned about stem cells therapies. But if you open the web, you can see huge variety of stem cells therapy, which is not regulated, but people are so excited about stem cells, the idea of stem cells, actually, that stem cells can cure you, they would go to any trial just in order to try to get some benefit out of it. And actually, I was really surprised a few years ago that Arnold Kaplan, one of the leaders of mesenchymal stem cells, said actually there are no stem cells. They are just mesenchymal cells that are therapeutic, they can secrete good things, and he even changed the name, therapeutic mesenchymal stromal cells rather than stem cells. So he said the stemness is not important at all. You can, if you get positive results, in most cases it's not because the cells are stem cells, because you inject them, they have some positive effect. Could be temporary, which is even better. And then if anything cells would do, where could you get the cells? So the placenta, which is a throwaway material or tissue, is a fantastic source. It has a lot of mesenchymal cells, and you just have to harvest them and prepare your preparation, and you can correct everything in the body. That's what people thought. And the notion really was accepted around the world, and you can see that people isolated these placental cells in different laboratories. The only thing that really intrigued me, why they tried to show that these cells can actually transdifferentiate to bone or cartilage, which has nothing to do with their activity. Because if you inject them, they don't make bone and cartilage. The best case, they just help the tissue regenerate itself. So the placenta, if we just look at it, is actually not one organ. It's two organs which are adjacent to each other. The internal one is actually part of the baby. The blood of the baby, of the fetus, is flowing through it, and it has very good connection with the baby. The external one is a very, very large tissue which feeds the baby, the fetal tissues, but they are not the same. But when people isolated cells, they took everything together, and then they injected them, and sometimes they got good results, sometimes worse. And if I was asked to talk about placental cells in Israel, so the whole area was led by a company that was called Pluristem. And since I knew that these cells are not stem cells, actually I rejected this company. That by the name you could see that they actually don't understand the material they work on, but they had fantastic setup in which they had huge incubators in which they could produce thousands of millions of cells, and in very good, I would say, regulated manner, and they worked with regulations, and they had 3 products which they presented everywhere. They called them stem cells. They were not stem cells, but when they injected them, they got some positive results. And then they came to me and said, you worked with cells and you work on radiation damages, so why won't you inject our cells and save mice from acute radiation syndrome, which means death, by just somehow injecting our cells? We want you to inject them actually intravenously and see what happens. I really didn't like the study, but I needed the funding. So I said, okay, I will have a first trial. So here I irradiated the mice, and of course, after 10 days, they began to die. About 2/3 of the mice died at that time, and then I injected the cells that they gave me IV, and to my surprise, I got some response. It didn't really save the animals, but I got some positive response. So I asked them to send me more cells. They said, we don't have these clinical cells, but we have cells that were not approved clinically. We can't tell you what they are, but they are mesenchymal stromal cells. And they tried them. I tried them. Suddenly I see fantastic results. This is almost 100% survival. And this is a preliminary study at that time. But where the cells, the mice, when injected IV, the mice didn't like it very much. They look a bit thick because all the cells went to the lungs. People expected them to go to the place where they are needed, but they went to the bone marrow, whatever. But they was stuck in the lung. And from the lungs, they had the effect. So I said, why should we? First of all, I want to get these cells. Secondly, I can inject them intramuscularly because everywhere I inject them, they have a remote effect. And that's what we did. And we got fantastic results. But after 2 injections, we had almost 100% survival versus a third survival in the controls. And after 10 days, something has happened. A few days after the injection, you can see even the weight of the mice was risen. We can see here, doesn't work. Sorry. Just, okay. Okay. You can see here that for some reason the light doesn't work here, but never mind. I'll use the arrow. Anyhow, you can see that the first days there is no difference between the animals that were injected or not. But the first 10 days, they are all alive. Suddenly, there's a peak of regeneration of the white blood cells, red blood cells, and the platelets. And you have to remember, injected placental cells and to the place into the muscle. And actually, they really regenerated all these lines of cells, which allowed these mice to survive, while the others who had no boost in the cells, in the blood cells, died. So we decided to see if it's a secretion effect, and together with the Charité, we set up an experiment in which we killed the animals at different time points after cell injections. And we found out fantastic results. We looked for human proteins in the bloodstream of the mice, and we could see that a lot of cytokines came out, some of them related directly to regenerative effect, like GM-CSF, G-CSF, and so forth, IL-8, IL-6. All this came out as a response to the radiation and the cells in the injection. Now in order to see that it's not the effect of the cells just by the injection, we injected them to non-irradiated mice. And in non-irradiated mice, we got no secretion at all. So it's a response of the cells that were somehow stuck in the muscles. They responded to the stress of the animal and secreted exactly the secretome that enabled the mouse regenerate. Nothing, no participation at all in the process, in the cellular process itself, just the secretory effect. And of course, we had the mice secretion as well, but this is not enough to save them. So together with the human proteins, we could save the animals. And when did it happen? Exactly when the ARS, acute radiation syndrome, killed the other animals. This was exactly the time points in which we had the adequate secretion. And we published all these works. So going back to Pluristem, And at that time, we presented them our data, and they had a new product now, which is called PLX R18, which is based on the fetal placenta, rather than the other products, which were mostly the maternal placentas. Now, why would the fetal placenta cells work better? Because the fetal placenta is supporting the baby. The baby has a stress signal which it sends to its own placental tissue, which secretes exactly the factors that are needed. So if there is a pancytopenia in the baby, it would be reflected by the secretory of the fetal placenta. But while the maternal placenta is just supporting the general, gives a general support to the baby placenta. So this company had a new product. But at that time, I disengaged with them and began my own study in which I could really control, see what I am isolating. I isolated the fetal placenta cells and expanded them by certain technologies that we developed. And we could show that they are fetal because the mothers were female. And by FISH, we could show that all the cells and the X and Y chromosomes, they came from the male embryo. And when we ran our experiment with much higher doses, we still got the effect. And even if we had splenectomized mice, they survived exactly the same, more than 80% survival after 8 gray exposure, which is the fully lethal dose. And you can see the bone marrow regenerating like a control versus the few residual animals that we managed to carry out to day 23, where we can see any bone marrow. And we published our results, and here it was really based on cells that you could follow up what they really were. Furthermore, we did a few experiments on other tissues. For instance, on skin, we irradiated the skin flap, which has without irradiating the mice itself, has no effect altogether. And we can see that on the mouse, and only on the irradiated area, and we can see that we get horrible wounds after some 50 days. And when we're injecting the cells locally, we can see regeneration. But the regeneration of the hair follicles gives us white hair because the melanocytes are not regenerating as fast as the hair follicles. And we could actually cut through the air and show here that we have almost full regeneration of the hair follicle. Of course, it could be used for other applications, but we haven't used it as well. So these placenta cells, I believe that they could also work for alopecia for other reasons, but this is still an open question. People ask me, of course, whether it should work as well. So the mechanism is very simple. I mean, what we could show that the cells are pro-regenerative. They trigger a secretion probably of relevant exosomes. And some people now think that such cell therapies induce transfer. I mean, the cells are full of mitochondria. They are very, very potent cells. If you inject them, maybe the mitochondria, which are affected by radiation, could be replaced, and this would be another mechanism that may be working. We haven't investigated it. So I have a short time, but I can tell you that we looked for other models like ALS, like syndrome in mice, in which we had anti-inflammatory effect only when we injected the cells intracerebrally, because when we injected them intramuscularly, the blood barrier didn't allow the stress signals to go down, or and the secretome to get to the brain. We had the IBD model, which showed us improvement of IBD. And we are now summarizing a model of diabetic rats in which we can see the regeneration of the pancreas very, very impressively. So I go back to Pluristem's product. So the product on the left were the product that were made from the maternal cells. And when I had my cells that they made into a product, I told them, please use them in order to work on the other model that you have. But they said, we are like a Titanic. We are clinical studies. We cannot turn left and right. We have to continue with what we have. But, of course, they ran the study for acute radiation syndrome, which with the cells that I found for them. And the clinical studies were not satisfactory for the FDA with the cells of internal cells. And surprisingly, which was like a bomb for them, and surprisingly, the FDA approved the studies that were done on human subjects with the cells against radiation. But at that point, the company was devastated because they invested so much in the other cells, which was a wrong decision to begin with. So they had to save themselves. And this is the epilogue. So they changed their name, and they had the facilities to grow cells, large-scale cell manufacturing, and now they are making steaks. And this is very, I mean, they saved some of the facilities, and they sell also all the incubators in which they grew the cells. But this is the end of the stem cells, placental stem cells story in Israel. So since the word placenta in Israel were mentioned, so I focused only on the studies that were done in my lab and by this company, Pluristem. And it gives us a lesson of how you really should treat data that you received. You have to have long breaths. You have to change things if you have good reason to believe that they are not working as good as you would expect. You shouldn't continue to run on something that you think there is something more optimal. And unfortunately, they failed in this direction. So thank you very much for listening. I just summarize what we've shown you, that xenogenic. By the way, I especially talk about the xenogenic. The human cells that were injected to mice were working fantastically. They were cleared away, which is good, because you don't want to inject cells that will stay forever, make tumors or whatever. You would like them not to differentiate, to do what they know to do, and eventually to be cleared away. The more active than the fetal cells were found to be more active than the maternal placental cells because, as I said before, the fetal cells talk to the embryo. They are responsive to stress signals, and that's why they work so well. And this just described the experiment that we've shown in my presentation. Thank you very much for listening for this short presentation. And whoever is offended by my approach to the other mesenchymal stem cells have to thank that they give me only 30 minutes, because I could give much more example how a whole field was actually people have working according to conceptions which are not relevant anymore. Thank you very much.
Rafi, thank you. Thank you very much.